So in the past, during my office hours, I had just kind of gone over the homework because that's typically what the test is going to be based on. I'm not going to be able to make it tomorrow, possibly, to the office hour review session or whatever, so I'm just going to spend a little while going through some of these questions. So number one, who was Gregor Mendel? Gregor Mendel was a monk who discovered Mendelian genetics, which is named after him. He was Austrian, I believe, and he worked right around the same time that Darwin was publishing on the origin of species. But he came up with Mendelian genetics, which is something that is very applicable even to this day. He didn't know anything about genetics. He didn't know anything about DNA. All he knew is that these plants that he was breeding had these characteristics. All right. What does phenotype mean? That is the actual physical characteristic that an organism will have. So, for instance, I have blue eyes. That is my phenotype. So, what does genotype mean? Genes are the actual genes that you have. Okay? So, many times genotypes lead to phenotypes. So, for instance, my genotype is that little b, which we will talk about in just a second. But that's my genotype, and that led to my phenotype. Genotypes don't always lead to phenotype directly. Many times there are environmental factors involved as well. So how are these different? One of them is a characteristic. Phenotypes are characteristics, whereas genotypes are genes. And again, genotypes don't always lead directly to phenotypes. Environment can play a role, ecology, lots of things can play a role, but very often they do. So what is a recessive trait? A recessive trait is a trait that if you have the dominant version, it's going to get covered up. And you should see this in the lecture I did on Mendelian genetics. But for instance, this is my brother's genotype for eye color. Let me zoom in a little bit, make this a bit easier to look at. There we go. So this is my brother's genotype. He has brown eyes because he has this big B, this dominant allele, and he has this little b, which is the recessive allele. So blue eyes is the recessive trait. Brown eyes is the dominant trait. And so I've kind of covered those. If you have a trait, if you have an allele and it causes you to have the dominant phenotype and the recessive allele is just covered up by that, that is called a dominant trait. So heterozygous and homozygous, I'm going to go back to me and my brother. So there's my brother's genotype. He is, what is he? I'll let you answer that really quick. He is heterozygous. Hetero meaning different. Zygous referring to zygosity, it's not important, but he is heterozygous. Okay, he has two different alleles. I, on the other hand, am homozygous. I have two of the same alleles for eye color. Okay, so I am homozygous recessive. So ask yourself this: what would homozygous dominant look like? You pause it or whatever, but homozygous dominant for eye color would look like this. Okay, draw and interpret a Punnett square for two heterozygous yellow pea plants. I refer you to page 198. Let's look at page 198. What am I talking about here? So, I mean, there it is. Just go to page 198. Make sure you kind of understand what's going on with that particular Punnett square. I'm not going to do it because it's just kind of right there. What is independent assortment? Independent assortment, this goes back to meiosis, and basically in meiosis, and you should go back to the lecture for this one, what happens in independent assortment is any version of the alleles that you get, it's kind of random. It doesn't make a difference which one goes where. So let me see if I can draw this. Where's my recorder? So let's say we're doing some meiosis, and we've got, let's see, how do I do this? Let's make it red. We've got this chromosome here, and we've got another chromosome over here. Okay, these are homologous chromosomes, like we talked about in week 4. It doesn't make a difference, and either of these can wind up in either gamete. Okay, so this could just as easily, this could just as easily be flipped around, where the blue one is going to wind up on the left, and the red one is going to wind up on the right. And then, once those things wind up moving over to their separate gametes, you can have a gamete that looks like this, maybe. That's one possible set of gametes. This circle, so this is clearly a gamete. Okay, this might not make very much sense to you if you haven't seen the lecture on this stuff, so please go check out the lecture. But just as easily as you could have this, you could have many other possibilities, especially if you're talking about the 23 pairs of chromosomes that we have. So maybe instead of having that combination of red and blue, maybe you have some other combination of red and blue. But independent assortment just states that the gametes can go into, it doesn't matter how your homologous chromosomes are going to line up. Okay, that is independent assortment. It's one of the laws of genetics. It's one of the reasons why there's so much diversity amongst people. Anyway, please check out the lecture for more information on independent assortment. I looked up the textbook definition. The textbook definition of independent assortment is the random distribution of homologous chromosome pairs on differing size of the metaphase plate during meiosis. Yeah, not super useful. Please check out the video lecture. What is a monohybrid cross? Well, that's when you're just doing a Punnett square of sort of one characteristic. It's still blue, but that's okay. All right, so let's put, we'll just say we're going to do my I allele, because that's one that we've talked about already. Here's my mom's I alleles. Okay, and here are my dad's I alleles. How do I know that these are what my parents' genetics are, by the way? You guys know how to figure that out? Like, how did I know that my mom was homozygous recessive, and how did I know that my dad was heterozygous? Well, I know for sure that my mom is homozygous recessive because I've met her, and she has blue eyes. And again, if you check out the videos, the only way to get blue eyes, check out the book, the only way to get this recessive trait is to have 2 recessive alleles. And how do I know that my dad is heterozygous? Well, I know that I have blue eyes, and assuming that, assuming that my dad is my dad, I know that he has brown eyes and that he has at least one little b allele because he had me. We'll talk about that. So let's look at the different combinations of children that my parents could have had. Okay, and you'll notice that half the kids that they can have are heterozygous, and half the kids that they can have are homozygous recessive. Okay, so let's say here's me, and one of these guys is my brother. Okay, so that's a monohybrid cross. A dihybrid cross, something I'm not ever going to ask you to do, but it involves doing a Punnett square of 2 different traits. And it just kind of gets, it's not any more complicated, it's just a little bit bigger. I'm not going to try to do one here because it'd be super ham-fisted. But yeah, dihybrid cross is just 2 different alleles on a Punnett square. Be very clear about that. Dihybrid cross is 2 different traits on a Punnett square. Okay. I repeated independent assortment. That's fine. What is an independent? No, spell checker. I know what I'm talking about. I know what I'm doing. Anyway, what is complete dominance? So complete dominance is like we were talking about. It's eye color. You either have one trait or the other. Now, obviously, it's not quite as simple as that because there are more eye colors than just brown and blue, right? but let's just pretend for a minute like that's accurate. Complete dominance is you either have one trait or you have the other. Incomplete dominance is, and again, check out the lecture, but the best example I can give for incomplete dominance would be like flowers. So let's white won't show up. So let's try that again. So let's say we have a flower here. Great flower, Aaron, nice job. Its alleles are going to be, we'll just say lowercase r, lowercase r. And we have another flower here. Great flower, nice job, Aaron. And we're gonna make it red, or I'm gonna attempt to make it red. Oh yeah, that's perfect. Brilliant. Okay. Its genetics are big R, big R. Okay, this is incomplete dominance. And when we cross these two, oh, there's not like a good pink. Yeah, it's kind of an okay. When we cross these two, we get a different phenotype. We get this like pink. We get this pink color. Okay, so in incomplete dominance, the heterozygous individual can be identified because it's a combination. It's a combination of the two phenotypes. This one's genetics are big R, little r. There's no reason that that's larger, by the way. I just didn't change the font back. But so incomplete dominance is you wind up with a combination of the phenotypes. Okay, this one is homozygous recessive. This one's homozygous dominant. This one is heterozygous. Homozygous recessive, homozygous dominant, heterozygous. So codominance, I'm going to answer the next two questions. And blood groups. Codominance is when you see both characteristics, both phenotypes at once. And I kind of discussed this a little bit ad nauseum in the lecture, I believe. But if you guys have heard of not genetics, if you've heard of blood types, you should know that you can. It's A, B, O. You can be type A, you can type B, you can be type O, or you can be type AB. And basically, what's happening here. Let's get a better color. Let's get a nice blood red. Basically what's happening here, here's a blood cell. They look like, sort of like, they're not as donut-y. They don't actually have holes in them. They have, they sort of like crease in the middle. These are terrible. These are really bad. Thank you for bearing with me on these. But you can have, you can be type A blood. And when you're type A blood, basically there's just a whole bunch of little flags on each blood cell saying, hey, I'm type A blood. Or, I should have drawn one more, but that's all right. You can be type B blood, which again, there's a bunch of little flags. There's more than 3 or 4, there's a bunch. On each blood cell that indicates, hey, this is type B blood, and your body accepts those. Or you can be type O blood, which you might think, hey, there's a bunch of little O flags on there, but really, there are no flags. Okay, but why this is codominance, and why this is kind of interesting, it's a thin blood cell for some reason, is you can have another thing happening, which is you can be type A, B, which means that each blood cell has a bunch of A flags and a bunch of B flags saying, hey, I'm type AB blood. Type AB blood. Okay, and so that is a great example of codominance because this type of blood has both versions fully expressed on it. It's not a combination of phenotypes. It is both versions being fully expressed. Okay. And how is this related to genetics? Well, you get it from your parents. In fact, here, let me do a Punnett square. Do a Punnett square of this. Okay. Draw on the screen itself. That's easier. Okay, so let's say your mom is type A. So what can her possible other allele be? Well, her other allele, she could have another A, or she could potentially have an O. Okay, O just means there's nothing there, so she could, if she was type AO, she'd have a bunch of A flags on her blood cells. If she was type AA, she'd have a bunch of A flags on her blood cells. Let's say your dad is type B. It's type BO. Haha. So what are the varieties of kids that a person who's type A and is heterozygous and a person that's type B and is heterozygous can have? Well, they can have a kid that is AB. Computer's lagging. They can have a kid that's type B. They can have a kid that's type A, or they can have a kid that's type O. And so you wind up with, actually this is a pretty interesting cross of folks. They can have every variety of phenotypic kids here. They're not having every variety of genotype, but they're having every variety of phenotype that they can have. Oops. Okay, so polygenic inheritance, multiple genes are having an impact on your phenotype. Multiple genes are involved in which proteins you're actually creating and how they're interacting with each other. And so for human beings, most of our characteristics are impacted by multiple genes. Okay, polygenic inheritance. How can environment affect inheritance? So there are many environmental factors that can cause you to express or not express different characteristics. Kind of glossing over that, but lots of stuff in the book, lots of stuff in the lecture. What is an X-linked recessive disorder? So let's start with what is X-linked. So X-linked disorders happen on your X chromosome. Okay, everyone has at least one X chromosome. If you have two X chromosomes, you are a female. If you have one X chromosome and one puny little Y chromosome, you are a male. So what is an X-linked recessive disorder? It is a disorder that happens on your X chromosome, and it is recessive. Very rarely will you see these in females, although it certainly does happen. So a great example of this, and one that I covered in lecture, is like color blindness. Okay, you will meet some colorblind women, some colorblind females, but for the most part, color blindness is much more prevalent amongst males. 3 examples of X-linked disorders. That's for you guys. What is an autosomal dominant disorder? So let's talk about autosomal. So autosomal, or autosomes, are your non-sex chromosomes. So you have 23 pairs of chromosomes. One set of those is your sex chromosomes. The other 2 are your autosomes. Okay, so autosomal dominant is a dominant disorder, a dominant trait that takes place not on your sex chromosomes. Aneuploidy, moving along, aneuploidy means that it is an organism with an incorrect number of chromosomes. So, incorrect number of chromosomes within the cell. By the way, you've almost certainly met someone who is aneuploid and not even realized it. Polyploidy, multiple, or there's some kind of duplication event or non-disjunction event of a chromosome. You have an extra chromosome in there. Non-disjunction is, if we go back to meiosis, when the homologous, or when the homologous chromosomes were separating, something went wrong. One of them didn't go to the place that it was supposed to go. Non-disjunction leads to aneuploidy. Okay? Non-disjunction leads to having the incorrect number of chromosomes. List and describe structural issues with chromosomes in the book. Those are in the book, so I will let you check those out. On to chapter 16. What is evolution? Evolution is a change in allele frequencies in a population. Change in allele frequencies, thank you Google, in a population over time. Or you could think of it as just being a change in a population over time. Okay, that is evolution. You should know it's a change, and you should know that it happens at the population level. This is sort of a microevolutionary definition, but it happens at the population level always. It's not an individual thing, it's not a community thing, it's not a biome thing. It happens at the population level. What is natural selection? Basically, in natural selection, you have a group wherein there isn't enough resources for every single organism to have some. Not all individuals can survive in natural selection. And then you have some heritable characteristic. You have something that a mother can pass on to her children, or a mother and father can pass on to her children. Maybe they're a little bit faster, maybe they're a little bit smarter, maybe they have a slightly longer dorsal appendage. Whatever it is, it gives them some evolutionary advantage, some survivorship advantage within that particular scenario. And what happens is those organisms wind up living better than the other organisms because they have an advantage. And so what happens is maybe that dorsal appendage gets longer and longer and longer. How important is evolution to the study of biology? It is incredibly important. It is sort of the central theme. Nothing makes sense except for in the light of biology. Darwin sort of came up with the idea of evolution. He studied it extensively, and he and a guy named Wallace wound up co-publishing a book about it. What was the name of the ship that Darwin sailed on? That's for you guys to figure out. Who were Darwin's influences? I'm looking for the 3 listed in the book, but more are fine. I'll let you guys study that one. Look that one up in the book. Who was Wallace? What were his contributions to evolution? Again, Alfred Wallace was this. He's also a biologist, was also very interested in why there were so many species and how they were changing, if they were changing. And he co-authored the book with Darwin. He was also a very important biologist. What did genetics have to do with evolution? Well, they have a lot to do with evolution. If your genes, if you have a characteristic that you're able to pass on to your offspring, it's going to be embedded within your genes. It's going to be in your genetics. It's a very fast answer, but yeah, check that one. Look that one up. Figure out maybe a better answer for this, but that's the basics of it. The modern synthesis of evolutionary biology is combining Mendelian genetics with the ideas of Darwinian evolution. And this is actually kind of like a 60 or 70-year-old idea. Now that we have even more modern technology, the modern synthesis is getting even better. The evidence is getting even more robust, even though it was already super robust in like the 50s. But yeah, combining Darwinian and Mendelian, remember Mendel from the last chapter, Darwinian and Mendelian ideas. Is there controversy over the truth of evolution? No, not within biology at all. There is absolutely zero controversy. Every biologist, I shouldn't say every, because as soon as you speak in absolutes, you're wrong, but 99%. Every biologist I've ever met, and I've met many, are convinced of the fact of evolution. List and describe the evidence for evolution. This is in the book. I will let you guys check that one out. I've got some repeated questions here. That's okay. What is a species? So this question is actually pretty darn complicated, and it depends on which species concept you use. The biological species concept is that if 2 organisms can interbreed and have offspring, and those offspring can have offspring, then they are within the same species. This is incredibly problematic, but for our purposes, we'll go with the biological species concept. There's also temporal species concepts, ecological species concepts, gametic species concepts. genetic and genomic species concepts. There are a lot, okay? So it's not quite as simple as you might be led to believe in a basic biology class, but we don't have time, so I'll leave it at that. On what level does evolution take place? It happens at, actually rewind this video and you can find the answer to this question. What is a genotype? Rewind the video and you can find the answer to this question. What is a phenotype? Rewind the video and you can find the answer to this question. What is a gene pool? A gene pool is sort of all of the genes within a particular group that can contribute to an organism or can contribute to the genetic diversity within an area. So you can think of it as being like all the alleles within a particular group. What is microevolution? The change in allele frequencies within a population over time. What are the 5 forces that drive evolution or microevolution? Well, you have natural selection. List these here. You got natural selection, genetic drift, we've got mutation, we've got gene flow, and actually I kind of want to put this one under here, and we'll leave it at, it's a sexual selection. Natural selection we have already talked about. Sexual selection is just kind of like a form of natural selection, really, but it's basically non-random mating. Basically, some members of the population are preferred over others. I'm going to gloss over this one for just a second. We'll come right back. This is the hardest one, so we'll come back to it in a second. Now we've got mutation, which is basically the change in alleles. So let's see, mutation, that would be like if this sequence changed into this sequence. Notice here that the G changed into a C. Okay, that is another driver of evolution. Gene flow, and actually I'll just leave this up here. Gene flow can be thought of as just migration. Genetic drift is the random loss of diversity. Or I guess I could say a random loss of diversity. This is in the lectures again, but basically what happens with genetic drift is you have a small population and certain alleles are removed for no reason. So for instance, if you lived in an anthill and a tree fell and killed half of the individuals living there, it wouldn't have anything to do with those particular ants. It would just be random chance, and that is genetic drift. What is genetic bottleneck? It's when a population is squeezed down like a bottleneck into a very much smaller size than where it started. You can think of it something like this. Let's say you start out, your population is big, represented by how wide this is, and then some event happens and causes your population to contract a huge amount. An example of this would be like the cheetahs in Africa. They were just kind of hunted down into a very, very small proportion of their original size, and there's very little genetic diversity left. And I think I said this in the lecture that I recorded in class and let you guys watch. But for cheetahs, no two cheetahs are more distantly related to one another than cousins, which is pretty wild. An adaptation is just a change in response to your environment. The biological definition of fitness means that you are able to reproduce more. If you're able to pass on your genes, biologically you are successful. Polygenic means multiple genes. Stabilizing, directional, and disruptive selection. So again, those are, I covered these in lecture, and they're in the textbook, and they're going to be able to draw you pictures that are better than the ones I'm going to be able to draw for you. What is the Hardyweinberg principle? The Hardyweinberg principle basically just states that if all of the ideas or all of the requirements for the Hardyweinberg principle are true, then evolution is not happening. So what are the requirements of the Hardyweinberg principle? Well, that is, no mutation is taking place. No migration is taking place. No natural selection is taking place, which means no sexual selection is taking place. And the last one, the most confusing one, is that it's a large population. Why a large population? Well, a large population prevents genetic drift from happening. By the way, what were our mechanisms, or what were the forces that drive evolution? There they are. If those things are not happening, evolution is not happening. That is the Hardyweinberg principle. Why is that interesting? Well, the Hardyweinberg principle is interesting because if you find that one of those things is not true, then you can go and pinpoint it and figure out, well, what is it that's driving the evolution of this particular species? Hey, evolution's happening here, guys. Why is it happening? What's causing it to happen? What is the biological species concept? I have talked about this already in this video. Do you think the biological species concept is always best for all organisms? Spoiler alert, it's not. I'll let you tell me why or why not. For the reason I think this is because the butterflies I studied in graduate school could definitely interbreed between species, but the genetics showed that they were actually quite different on a genetic level. Anyway, what is speciation? It is one species turning into a different species, or one population changing into a different species. Allopatric speciation means happening in different areas. It's basically like the classic example of this would be like, okay, an earthquake caused a river to move and then suddenly your population is split in two. That would be allopatric speciation. Speciation that happens in different areas. I'm going to let you guys go through the book and find the reproductive isolating mechanisms to define. I'll define it, I suppose. A reproductive isolating mechanism is just something that would prevent two organisms from interbreeding with one another. Okay, so what might prevent two things from interbreeding with one another? Think about like a mechanical one, like a Chihuahua and a Great Dane. Okay, hopefully those two. My God, I didn't think about that. Hopefully those two never interbreed with one another. And I'll let you list three of those. Think about it. Get three of those down. What is sympatric speciation? It's speciation that happens within the same area. And so, like, I'll give away one of the reproductive isolating mechanisms. Let's say you're a bird and you live in a tree and you get up early in the morning. You're never going to see another bird of that same species that doesn't get up until late at night, right? So that's an example where evolution can take place because there's no reproduction happening between those two species, even though they're sympatric, even though they're in the same area. And again, stupid autocorrect. I know what I'm talking about. It's anadaptive radiation. And adaptive radiation is what happens when you introduce a species into a set of new niches. So an example of that would be like the butterflies I studied. They got to North America 3 million years ago, and they found a whole bunch of area here that they could just spread out onto that had their food plant pre-existing for them. I'm going to let you guys figure out this binomial nomenclature stuff, but it is just genus, species. Humans are Homo sapiens. Okay. Systematics, yeah, that's one you can look up in the back of the book. Taxon, it's a little bit more complicated. A taxon is just kind of any grouping that you can think of. So, like primates are a taxon. Homo, our genus, is a taxon. Okay, our species, sapiens, is a taxon. It's any of those particular groups. A phylogeny is just basically, it's a picture of how different things, different organisms are related to one another. Coevolution is evolution that happens at the same time. A fantastic example of this would be if you look at large flowers and their hummingbird pollinators. Okay, I know I'm talking fast. I'm just really hungry. I put the horse before the cart here. I should have put convergent evolution like up here. Coevolution, again, it's what happens when two things evolve with one another. I think the example I gave in the lecture was I was talking about newts that are quite toxic and snakes that are resistant to that toxin. Okay, that's coevolution. They've evolved together. Convergent evolution is when things live in similar environments and so they wind up with similar characteristics. The best example of this would be dolphins and fish. Hopefully you guys are aware that dolphins are mammals. Or bats and birds. Hopefully you guys are aware that bats are also mammals, but they fly around, right? They live in similar environments, and so they wind up having these analogous traits. They are traits that are similar to one another, but they are not from common ancestry. So analogous traits, traits that are similar to one another, like the fins on a dolphin and the fins on a fish, but are not from common ancestry. Homologous traits are traits that are similar to one another because of common ancestry. So the fact that if you dissect a pig, it has basically the same internal layout as a human being, that is a homologous trait. The fact that if you look at like a chimpanzee and you see, hey, they have like some of the same features as us, that's because of homology. Those are homologous traits. They are from common ancestry. Oh, thank God we're done. It is dinner time. I'll start recording the other video in a minute.